Researchers have broadened the genetic alphabet to develop novel proteins. Greetings to everyone. Today's video centers around a fundamental principle taught in every introductory biology course. Proteins are made up of combinations of 20 distinct amino acids organized into various sequences akin to words. However, researchers at Scripps Research have introduced an innovative method that utilizes 4 RNA nucleotides instead of the conventional 3 to incorporate non-canonical amino acids into proteins. This technique facilitates the precise integration of new amino acids without the need to modify the entire genome, thereby enhancing the ability to create proteins with specialized functions for use in bioengineering and drug discovery. By simplifying the process and expanding the range of usable amino acids, this approach opens the door to groundbreaking small molecules with considerable applications across multiple fields. Now, without further delay, let's dive into our discussion. Researchers at Scripps Research have established an innovative framework for the straightforward incorporation of non-canonical amino acids into proteins. This method, detailed in Nature Biotechnology on September 11, 2024, centers on the utilization of 4 RNA nucleotides instead of the usual 3 to encode each novel amino acid. Our objective is to create proteins with customized functions applicable across various domains, from bioengineering to drug discovery, states senior author Ahmed Badran, PhD, an assistant professor of chemistry at Scripps Research. This innovative method allows us to integrate non-canonical amino acids into proteins, bringing us nearer to achieving that objective. To generate a specific protein, a cell must convert a strand of RNA into a sequence of amino acids. Each amino acid is represented by a codon, which consists of 3 nucleotides of RNA. However, several amino acids can be associated with multiple codons. For example, the sequences UAU and UAC in RNA both signify the amino acid tyrosine. The role of small molecules known as transfer RNAs, tRNAs, is to connect each amino acid to its matching codons. Recently, scientists have developed methods to reassign a codon with the goal of incorporating entirely new amino acids into a protein. For example, by altering the tRNA associated with the UAU codon, it could be interpreted by the cell as representing a different building block rather than tyrosine. However, to avoid the unintended integration of the new amino acid into numerous other proteins, every occurrence of UAU within the cell's genome would also need to be changed to UAC. Badran states, utilizing whole genome recoding to generate free codons can serve as a potent approach. However, it presents significant challenges due to the extensive resources needed to construct new genomes. Additionally, predicting the impact of these codon modifications on genome stability and the production of host proteins can be quite difficult for the organism itself. Badran and his team aim to develop a streamlined plug-and-play approach that would integrate selected non-canonical amino acids into designated sites within a target protein, all while preserving the cell's standard biological functions and avoiding the need for comprehensive genome editing. This required utilizing tRNA that had not been previously designated for any amino acid. Their answer was a 4-nucleotide codon. The researchers were aware that in certain instances, like bacteria rapidly developing resistance to drugs, 4-nucleotide codons had evolved naturally. In their latest study, they examined the factors influencing cells to utilize a 4-nucleotide codon instead of a 3-nucleotide one. Their findings revealed that the sequences surrounding the 4-base codon played a crucial role. Commonly used codons improved the cell's ability to interpret a 4-nucleotide codon for the incorporation of a non-canonical amino acid. Badran's team subsequently investigated the possibility of modifying the sequence of a single gene to include a novel 4-nucleotide codon that the cell would correctly utilize. Their approach proved successful. By surrounding a target site with commonly used 3-letter codons and ensuring adequate levels of the 4-nucleotide tRNA, the cell was able to incorporate any new amino acid linked to the corresponding 4-letter tRNA. The research group conducted the experiment again using 12 distinct 4-nucleotide codons and applied the technique to create over 100 new cyclic peptides known as macrocycles, each containing as many as 3 non-canonical amino acids. Badran states, "These cyclic peptides evoke the bioactive small molecules that can be encountered in nature. By leveraging the programmability inherent in protein synthesis and the variety of building blocks available through this method, we can develop novel small molecules that will offer thrilling possibilities in drug discovery." He notes that this new technique for incorporating non-canonical amino acids is simpler than earlier methods, as it requires changing only one gene instead of modifying the entire genome of a cell. Furthermore, the potential for using additional non-canonical amino acids in a single protein is greater due to the existence of more 4-nucleotide codons compared to 3-nucleotide codons. Badran states, "Our findings indicate that non-canonical amino acids can now be seamlessly and efficiently integrated at various locations within a broad range of proteins." He adds, "We are thrilled about the potential these opportunities hold for our current research and to offer this capability to the wider community." He points out that this technique may be applied to modify current proteins or to develop completely new ones, which could be beneficial in various fields such as healthcare, production, and chemical detection. This research received financial backing from several sources, including the National Institutes of Health, DP 50D024590, the Research Corporation for Science Advancement, the Sloan Foundation, G2023-19625, the Thomas Daniel Innovation Fund, 627163_1, an Abdul Latif Jameel Water and Food Systems Lab Grand Challenge Award, GR 000141S 6241, a Breakthrough Energy Explorer Grant, GR 000056, the Foundation for Food and Agriculture Research, 28000578, a Homeworld Collective Garden Grant, GR 000129, the Army Research Office, 81,341 BBECP, the Hope Funds for Cancer Research, HFCR 230301, a Skaggs Oxford Scholarship, and a Fletcher Jones Foundation Fellowship. To support our channel's growth and ensure broader awareness, kindly hit the like, share, and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.